Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thanks very much. And the last words were really important, namely, I left a lot out. We also left a lot out, as you will notice in this, what I'm going to present here. It's some afterthoughts on work that I did with Luca and Andrew really started 10 years ago, so it's another anniversary that we have here. That was the period which actually connects to the description that Ehud Shapiro gave on his work with his PhD student, Aviv Regev, because her work also stimulated our interests. We got in touch with her, found her thesis. She sent it to us at the time when things were not yet all published. And this stirred our interest in this. And then we got into contact with Luca. Andrew was also just arriving. You still hadn't finished your PhD thesis. And we were starting to play around with approaches that were of interest to us, namely to understand the dynamics of gene regulatory networks. So that's the work that we then published in 2006, which gives an introduction to how to use the stochastic pi calculus for the description of these networks. In part, the work is, of course, motivated by, at that time, really novel work by Stan Leibler and Michael Elowitz, who had this famous paper on the repressor, which is a synthetic gene circuit in which three genes are made to interact through a combinatorial swap of the promoters, such that, as in the terminology that Attila used today, a ring oscillator is generated in which each of the genes is repressing one of the other genes, so that this kind of circular thing arises, and then, of course, there is some reporter gene. So the question is, how would you describe such a simple network which you can realize in bacteria in a plasmid? And so being a physicist, so this is a collaboration not of a biologist and a computer scientist, but of a physicist and a computer scientist. So it's like people who are both not don't have the perfect biological vision, at least not at the time. We were really trying to make sense by way of abstractions. And one way of abstracting is, of course, the programming language that we used, the pi calculus. But the other abstraction is, of course, how do you model this gene? Because, of course, you can say the gene dynamics, I can take account lots of molecules that may intervene. But being a physicist, you're, of course, motivated by, say, Occam's razor, or Einstein's version of it saying you should simplify, but not too much. So the question is, how far can we simplify? And the model that we made essentially is based on this small number of reactions. The first is a basal transcription. So we take a gene as the prime actor, which is producing an output B, which we can interpret as an mRNA or, as we did directly, as the protein itself. So we ignore the whole level of mRNA. It's sort of shifted in the transcription rate. Then there is a transcription factor binding reaction. That is, a protein A can interact with the gene with a specific rate and put the gene into an active state or an inactive state. And it's immediately releasing the protein again. This is a controversial reaction. I will come to that. And then we have two possibilities. This sort of activated or, say, enhanced gene then has two possibilities. It either relaxes back to its original state by producing new protein. That would be really the activating process. Or it's a repressing process in which this gene is just doing nothing in state G prime and then returning at a rate eta back to state G. And then, of course, the proteins A and B can be degraded. So I already mentioned here two notions, pos and neg, that will come up on the next slide, because that's the way how, the upcoming slide, because that's the way that these genes have been represented then in stochastic pi calculus. In fact, what we do is we write down processes that here, this is directly copied from our paper at the time, where in fact all interpretations are already written, so I really don't have to read them. Essentially, the gene is represented by its product, which is the transcription factor that is produced. There is a stochastic delay, which simply means the simulation is being done with the underlying Gillespie algorithm. And so this rate is essentially reflects just the waiting time until this particular reaction occurs. And then there is the parallel process, again, of this transcription factor that was explained later, and the gene, the original gene, the initial state. The transcription factor indeed corresponds to an output, on a channel B, goes back to the original state, and then there is a choice between either this production or a degradation with a corresponding degradation rate, or waiting time in this case. And then, based on these two underlying processes, you can build two essential elements for our gene networks. This is the negative gate, so it's negatively regulated, it's repressed, or it's the pos gate, which is positively regulated, so the output is activated. And this is, again, then just a composition of these processes before. So this is an input on A, there's an inhibition delay, and then the process goes back to itself. And then there is the combination of the constitutive transcription and going back to the initial state. The same here, but here, of course, it's a little bit more complicated, because then, of course, you have the two possibilities of output. So I noticed actually yesterday morning that the notion of pos and neg has a long history, because when Gordon Plotkin showed your PhD work, these specific terms were already mentioned in the Stick and Stones, where they certainly had a different meaning, but it was just apparent for me. So once we have this, we can then start building small gene circuits just by sort of compositionally composing these genes. And here I only drew those that are sort of repressing, but of course you can do the same family with the activating genes. So it's like a self-regulating gene, something which will actually turn out to be a bistable system, so two mutually repressing genes. Then the famous repressor with the three genes that repress each other in a circular fashion. Or, of course, the second class would be you have sort of linear chains of genes that are repressing each other downstream or activating. So the repressor is this, with a marked error that I detected, because this is not the repressor. Here should be an A, because indeed now the nomenclature, the definition of the gates is such that you can immediately see what's happening here. A is input, B is output. Output B is input here, then C is output, is input here, and then in order to close this properly, this should be an A. So the way it's written here, it's more like a modified bistable system. So this is the output. It produces a beautiful oscillation. This is the protein number as a function of time of this circuit. And this is no surprise, because that's what the repressor really does. So qualitatively, that's OK. But it nevertheless posed a problem, because why did the thing oscillate in the first place? What actually makes it oscillate? The version of the repressor that was published by Stan Leibler, they also did simulations or computations beforehand, but they only worked in a deterministic setting. So they wrote down differential equations for the protein concentrations. And in these equations, one typically puts, for good reason, what's called a cooperativity, a Hill coefficient, into these equations. So there is a nonlinearity. And in fact, that nonlinearity is needed. It corresponds essentially not to just having monomers of proteins that act as transcription factors, but rather dimers or higher order structures. Because only in a deterministic setting, only if I have this nonlinearity, then the system actually too has states of, each gene has a state of low and high protein concentration between which it can switch. So it needs some sort of bistable, at least the possibility of having a bistable state. And the nonlinearity guarantees this. But in our stochastic simulation, we were only considering monomers of proteins. We neglected the dimerization step. Of course, we can put it in. It's just one more reaction. There's no problem with that. But the thing oscillates already without any cooperativity. So the question is, why does that happen? And then this was a thing that at the time we sort of had some ideas about, because indeed in the stochastic system there is always a possibility that the system can go to a state of no proteins. And then it can sort of revive again. The state of no proteins is not accessible in a deterministic system. So that's indeed turns out to be also the quantitative correct interpretation, but we can now be more precise about it. And then after the work that we did with Luca and Andrew, we published 2 papers that Andrew showed on this in 2006 and 2008. And I had 2 postdocs that continued our work a little bit, and I just indicate briefly what are the main outcomes of this. Because in one of the papers, what we did some more really detailed comparisons between the stochastic system. So we see here now not the protein concentrations as a function of time, but the phase diagram, so to say, in which we have plotted only the concentration against each other. And so you see this sort of fuzzy limit cycle that this produces. And in the deterministic setting, you see indeed a beautiful limit cycle oscillator. But how do we get to a deterministic system from our stochastic reactions to begin with? Well, I'm hiding this because this is, of course, a technical step. There is no one unique way of deriving deterministic equations. But one way would be to say we look at an average over a cell population that is synchronized. And then we can write down equations both for the gene and for the protein concentrations. And indeed, the outcome, the stability, and the degree of nonlinearity that's needed in order to have a limit cycle very much depends on the details of the dynamics. That means if I take only the protein concentrations as dynamical variables and ignore the gene, then I need a higher cooperativity than if I take the genes as proper variables themselves as well. So they indeed increase the phase space available for the limit cycle behavior. Yeah, this 2-gene circuit that I showed before is indeed already a properly bistable system. That was noticed at the time also when we worked on the repressor later, and with different methods. So this is the calculation again with the pi calculus, but other people have noticed that as well. So indeed, this is a fundamental feature. Without cooperativity, you can have a bistable system again. You would not have that with a deterministic system. That's really a feature of the stochastic system. And then what's the solution? Well, with another postdoc, we finally did something very detailed because we just looked at the self-regulating gene in both its repressed and its activating version. I'm skipping over all the technical details because we could solve actually the master equations for this system exactly. And that means you have access to the full probability distributions in the whole parameter spaces. And that really pinpoints then what happens here for a particular set of parameters. You can see that the probability distribution as a function of protein number indeed is bimodal. It has a minimum, has a maximum at a specific concentration of proteins or number of proteins. And it always has a maximum for a particular, can have, let's say, at 0 protein number. And why is that always at 0 protein number? It's never at a finite protein number. So it's never something which has 2 maxima at finite protein numbers. That's indeed a consequence of the peculiar choice that we made of our reaction, namely this one, which has always created a fight with referees because it's clearly an abstraction in the sense that there is no transcription factor complex that exists it exists only for 0 time, because it's like a reflection, you can say. This is like the protein interacts with the gene and is immediately reflected off the gene and is transformed into an activated state. So we have no complex between the transcription factor and the DNA, which of course is there in reality. And the consequence of this is indeed that in our model, there is no maximum at finite protein number at low concentrations. But there is always the possibility of having this bimodal structure. And that means that the feature of bistability is always there. And this guarantees that in these stochastic simulations, already the oscillations can appear. So that is the short story. This is an afterthought to this model. We now have not understood much about biology, but we have understood something about the model that we put in. And I think that's the main message here, is to try to, if one makes abstractions, of course I can say I represent biological reality in all detail, but then I make a model that's as complex as biology itself. So I have to cut down in terms of the actors of this system that I represent. And this is a way to say really quantitatively, within this abstraction, what are the properties that I can build in order to get the behavior of the system qualitatively correct. With this, I like to thank Luca and Andrew for a very nice collaboration that we had for a couple of years before we sort of parted in different directions. I then also went into much more complicated biological situations. Namely, I worked in recent years on regulation, gene regulation in eukaryotes, in particular level of chromatin. And I'd also like to thank my postdocs, Claudiu and Yves, for their work they put in to polish up the final questions a little bit in this model. And of course, I thank the organizers, and happy birthday, Luca.